Three. Hello, everybody. Welcome to another WBESS episode. For those of you who are new to this series, WBESS stands for Western Biology Evolution Seminar Series. I am your host, Matheus Sanita. I'm a PhD student in biology here at Western University. Today is our 17th episode, and I have here with me Jess Peers, who is soon to defend her PhD. She'll walk us through her PhD work in population genetics of cheetahs. So, Jess, please take it away.
Hi everyone, I'm excited to be here and talk about my PhD work today, focusing on the effects of low effective population size, focusing on the cheetah. So I'm going to start with a statistic that I'm sure is not new to many people. So many species globally are threatened with extinction currently. We're facing a mass extinction. And that's 28% of all assessed species and 27% of mammals are threatened with extinction. And one of these species is the cheetah. So the cheetah is a mammal found across mostly Africa, and also there's a few small populations in Asia. So as you can see on this map here, they're found around Africa, but in very small, fragmented and isolated populations. And this can have a negative effect on the population genetics. So population decline can lead to genetic bottlenecks and increased inbreeding. So a genetic bottleneck is where you may start with a very diverse population, which goes through a bottleneck, which may be due to disease or climatic conditions or poaching, any effects like that that reduce the population size rapidly. And although the population size may then recover, it's got a lot less diversity in it than it did originally. So this population has reduced genetic diversity and also an increased risk of inbreeding. And it's also prone to genetic drift, which is the increased risk of fixation of weakly deleterious mutations. So these are harmful mutations that aren't negative enough to prevent the animal from mating and passing on its genetics, but still have a negative effect on the fitness of the individual. And these can accumulate in small and inbred populations more than they would in larger diverse populations. And this can lead to inbreeding depression. And these are the phenotypic changes we see due to this accumulation of deleterious mutations. And this has been observed in many species that have historic bottlenecks. The cheetah is one of these, but also the black-footed ferret, sea otter, pink pigeon, kakapo. Those are some of the most widely focused on species, but there are many more species that have had bottlenecks and symptoms of inbreeding depression identified. But there are some common symptoms across these species that are likely due to inbreeding, such as disease susceptibility, reproductive defects, and developmental defects. And in fact, the cheetah has had severe genetic bottlenecks historically. So it had a severe genetic bottleneck around 10 to 12,000 years ago. And it's likely that they've had a continued decline since then. And because of this, they've had a low effective population size for a long time. So this is the number of individuals passing on their genetic material in the population. And this number is very low in the cheetahs. And because of this, they have many observed symptoms of inbreeding depression. And cheetah populations are continuing to decline. So there have been observed reproductive defects in cheetahs. They have quite poor quality sperm. Sperm motility is very bad. And this means that their reproduction is not often successful. They have developmental defects that can lead to high infant mortality. They have asymmetrical skulls and also kinks in their tails and immune system issues. So they're more vulnerable to particularly respiratory infections. And as I said, cheetah populations are continuing to decline, which may mean that cheetahs are running out of time. So today I'm going to talk you through the three main projects I've worked on in my PhD to look at the impacts of this inbreeding in cheetahs. So firstly, I looked at premature termination codons, or PTCs, and whether these are contributing to inbreeding depression symptoms in the cheetah. I've then looked at using machine learning to predict functional non-coding regions and then look for deleterious mutations in the non-coding genome, particularly focusing on using machine learning in non-model species. And then I've looked at deleterious mutations in a wider set of population data and applying this to conservation. So thinking about the distribution of these deleterious mutations across wild and captive cheetah populations. So firstly, looking at premature termination codons, or PTCs. So the genome is made up of coding and non-coding parts, and I'm going to focus firstly on the coding parts of the genome. So these are regions that encode a protein. And mutations in coding regions can impact the protein that's made. So a mutation in the DNA can then lead to a change of the amino acid in the protein, and this can change the function. But there are certain mutations where this can in fact lead to a stop codon earlier than would be expected, and this truncates the protein, and it's likely then decayed through nonsense-mediated decay, and the protein isn't carrying out the function that it should carry out. So this can be quite deleterious. So I wanted to look at genes that had these premature termination codons or stop codons in the cheetah. So previous studies in the cheetah had done comparative genomics with a handful of felid species. So these are all of the Felidae species here, and the previous studies included species with the black arrows. So as you can see, there's quite a large gap between the cheetah, which is in light blue here, and the most closely related species included, which was the domestic cat. So this means that the mutations identified in these studies may not be specific to the cheetah and may have occurred somewhere else in the lineage between domestic cat and cheetah. They could be specific to the whole puma lineage, for example. So what I wanted to do was add in additional species that mean it was possible for me to tell whether the mutations I've identified are specific to the cheetah or not. So I added in the puma and also a couple more felid species that genomes have been published for, but this enabled me to confirm that the mutations I identify are cheetah specific. And the other thing that these previous studies hadn't done was they'd both focused on deleterious mutations as a whole, but not pseudogenization, which is what happens to a gene when there's a premature stop codon. So both these studies found really valuable results about particularly an increased amount of mutations in sperm-related genes, but there was still a gap in focusing on genome-wide premature stop codons. So to tackle this, I started off by extracting coding sequences from 12 mammal species. So I had the 8 cat species that I showed you before, and also 4 mammal outgroups. And I ran a tool called GeneSeq to Family. So this is used to identify gene families and generate gene trees like the one you see here. So I gave all of the coding sequences to GeneSeq to Family, and it gave us output gene trees. And it tried its best to put one gene in each gene tree. This wasn't perfect, but generally it would separate gene duplications. And I could then use these gene trees and a tool called Notung to carry out gene tree reconciliation and identify gene trees where there wasn't a copy of the cheetah gene. So I took all of these potential gene losses and then did a lot of manual validation and curation of these. So I looked at things like comparing the cheetah and puma sequences to confirm that the mutations were specific to the cheetah and not shared in the puma. I checked start codons to confirm that it wasn't a misannotation problem, and also looked in genome gaps. So the cheetah genome that I was using was the original cheetah reference genome that came out in 2015, and that's a short read genome. So there were some gaps in that assembly, and I wanted to check that these gene losses weren't just missing because they were falling in a gap in the genome annotation, genome assembly, and also whether there or not they were missing because they weren't annotated. And I ended up with 65 genes with novel premature stop codons that I was very highly confident in. So I then had a look at adding some available population data to see whether these genes were shared in more than one cheetah. So as I said, I was using the original cheetah reference genome for this, which was from a cheetah called Chewbacca. There was a short read genome that came out in 2015. And alongside this publication, they also resequenced 6 individuals. So I used those sequences as well. And as I was doing this work, a long read cheetah genome came out. So I included this as an individual in my population study too. And what I found was out of the 65 genes, about 24 of them didn't have enough coverage for me to call population data. Of the ones that did, I found 22 of these genes shared the novel stop codon in the same gene in the same position in the resequencing data as well. And then 4 genes also shared the stop codon in the long read genome. So this gives me confidence that these stop codons are real and are shared across more than just one individual. So thinking about why 22 genes were shared in the short read and resequencing data, but only 4 were shared when I added in the long read. The short read genome and the resequencing data are all from wild African individuals. So the genome was from Namibia and the resequencing data was from Namibia and Tanzania, whereas the long read genome was from an individual in Lisbon Zoo that had been in captivity for at least 4 generations. So it's possible that the captive breeding that the Lisbon Zoo individual has been exposed to has potentially removed some of these deleterious mutations, whereas the wild individuals are still in small fragmented populations, and so those mutations have persisted. So just looking at what those genes actually are, the 22 genes shared in the African individuals were primarily involved in male fertility, but also had functions in immune system, developmental defects, and cancer. So you'll remember most of these are symptoms that are associated with inbreeding depression. So these mutations could be contributing to the inbreeding depression symptoms we see in the cheetah. And the 4 genes that had novel stop codons were associated with reproductive defects or immune system issues. And one of these in particular, CCDC189, a previous publication in cattle has found that a spliced donor variant in this gene is associated with a sphenospermia, so poor sperm function in a species of cattle. So I then looked at the mutation that I had found and where it falls in the gene compared to the mutation they were looking at. So this is a figure from my publication that you can find on this QR code. But what we see is the mutation I find falls in between 2 of the subunits that are primarily involved in the function of this gene. So it's likely that the mutation I find is truncating at least the coil domain from this and therefore impacting gene function. And it could very well be causing similar effects in the cheetah as it had done in the cattle. So next, I'm going to talk to you about my work on using machine learning in non-model species. And this was to predict functional non-coding regions. So I've covered my work on the coding genome, and these are regions that code for proteins, but there are also functional non-coding regions like enhancers and promoters that impact gene expression. And these don't necessarily change the protein themselves, but can impact how much of the protein is expressed. So the majority of disease and trait-associated GWAS variants have actually been found in functional non-coding regions. So, for example, canine OCD is associated with a couple of variants in a highly conserved regulatory element. So these regulatory elements contain a lot of important variation. So it's important that we look at them. But this hasn't been done previously in the cheetah. And this is because functional non-coding regions like enhancers and promoters are more difficult to find than genes. And there's also a complicated relationship between the regulatory regions and the genes that they regulate. So as you can see here, rather than just taking the gene sequence, you've got a 3D structure that you need to consider, and the promoters and enhancers are upstream of the genes. So we've previously relied on experimental data or sequence conservation to identify these regions. But experimental data is not available for many non-model species. You need high quality samples, quite complex experimental protocols, and this can be expensive and time consuming. And also it can be difficult to get these samples in the first place. And sequence conservation, which has been used where experimental data isn't available, is not always an accurate predictor of function. So just because a DNA sequence is conserved, this doesn't mean the function is conserved. And similarly, the function can be conserved even if the sequence isn't. So to solve this problem, I wanted to use machine learning to try and identify these functional non-coding regions like promoters and enhancers. So my aim was to apply deep convolutional neural network approaches using ATAC-seq data from model species and to predict functional non-coding regions in non-model species to then be able to identify deleterious variants in these regions. So I used a tool called Explain, which I'll explain in a moment, and took ATAC-seq data from BarkBase and the ENCODE project. So Explain is a tool that takes in a DNA sequence and one hot encodes it. So it turns it into a table of ones and zeros that a computer can understand. And this is what the model is trained on. Now, Explain stands for Explainable Neural Network. So it's based on a neural network approach, particularly convolutional neural networks, but it also contains elements of linear models. And what this does is makes it explainable, hence the name. So rather than it being a black box of you input your training data and it outputs a prediction and you don't know how it's made that prediction, Explain actually gives you the filters that it's used to make its decisions. So it's also an adaptation of neural additive models. And what this means is it runs multiple convolutional neural networks at the same time and then combines this information together to make its predictions for the output layer. So I trained this on ATAC-seq data. So ATAC-seq sequences open chromatin, and this is used as a proxy for functional non-coding regions because if the chromatin is open, it's accessible to transcription factor binding, and therefore transcription and translation. So I downloaded FASTQ files from Barkbase and ENCODE for the human, mouse, and dog. I selected the tissues of heart, liver, and stomach. This was because these were the tissues available for all of these species. And I wanted to include multiple tissues so that I wasn't biasing my predictions to anything tissue specific. So I processed this data using the Nextflow NF-core ATAC-seq pipeline and then called narrow peaks for each species. So I took regions that had a high volume of reads in the same place, and rather than calling wide peaks, I took narrow peaks that were 201 base pairs long. So these were my training data sequences. So to train the model, I combined all of the tissues together. Like I said, I didn't want anything to be tissue specific. And then I took all of the training data from chromosomes 2 onwards and separated this from the data from chromosome 1. So I trained each model on all of the chromosomes except chromosome 1 and then tested independently on the chromosome 1 data set. And this gave me an independent test set to confirm that the model wasn't overfitting and that it was performing well. And the way that I measured model performance was through precision recall. And this is essentially a way of measuring how well the model is performing, whether it is being precise in its predictions and whether it is recalling as many positives as possible. And what I found was that within species, the model performed the best, as you would probably expect. So when trained on the human and tested on the human, model performance was much higher than when trained on the mouse and tested on the human, for example. But as you'll notice, my axis here goes from 0.84 up, and actually precision recall around 0.9 is generally considered very good. So although it looks like there's a big difference here, all of these models are performing quite well. But it's still interesting that there is a disparity between training and testing on the same species compared to different species. Obviously, in the cheetah, I don't have training data. So the point of this was to find a way of training on one species and testing on another species and having maximum precision recall. And what I found was when I trained on more than one species and ran it on a different species, model performance improved. In fact, what you'll notice is that the model that was trained on the mouse and the human and tested on the dog actually performs slightly better than the model trained on the dog and then tested on the dog. So this gave me confidence that I can transfer this from one species to another and therefore use it in non-model species where we don't have training data. The problem that I found though was unbalanced data. So the genome is very large and the functional regions of non-coding DNA are very small. And in these model species, I know where the functional regions are. So I could balance out my training data. I could take the same amount of real functional regions as non-functional regions and use this to train the model. But in the cheetah, I don't know where these regions are. So I have to just use the entire non-coding genome and ask to make predictions there. And what I found the issue was with this when I tested it in the dog was that the performance on balanced data sets was around 0.9, but in imbalanced data sets, it fell to around 0.1. And what's happening here is that when I normalized the data for the number of positives and negatives, you can see that the model is still performing well. So over zero point 0.5, the model is more likely to predict something as a positive than a negative, which is great. That's what I want to see. But when you look at raw counts alone, there are so many fewer positives than negatives that you can barely even see the positives here. And this is what's skewing the precision recall score. So I then had to think about ways that I could filter out false positives. Um, so I was sure that the precision recall scores were low because of this data imbalance and needed to add additional filters to reduce the number of false positives. And as I said, I tested this in the dog, and this was because I was using the dog as a kind of fake non-model species. I had the least amount of data available in the dog, but I was using my human and mouse model that I trained and applied to the dog to but because I know the answer in the dog, so to speak, I have data in the dog, I could use this to confirm my predictions. And then I can apply this to when I'm predicting the cheetah where I don't know the true and false positives. So for the next few slides, I'm going to use the phrases true and false positives and negatives. And what I mean here, if you're not familiar with these terms, is a true positive is something that was predicted as a positive and overlaps with an ataxic peak, so is a positive. Whereas a false positive was predicted as a positive, but isn't a positive. And the same with the negative. So a true negative was predicted as a negative and is a negative, and a false negative was predicted as a negative, but isn't a real negative. So the problem I had was a large number of false positives that were predicted as positives but aren't real positives. And I needed to find ways of filtering these out. So the first thing I did was look at the explain prediction score. So explain gives me a score between 1 and 0 for each sequence, and that's telling me how likely it is based on explain's learning of regulatory regions, how likely that region is to be a real regulatory region, how likely it is to be a positive. And I did find a significant difference between the true positives and the false positives here, in that the explain score for true positives was generally higher than for false positives. So when I filtered the explain score for 0.75 rather than just taking anything above 0.5, this filtered out quite a lot of false positives. But I still had a ratio of true to false positives of 1 to 17, which was higher than I would have liked. So I had to look at other ways of filtering as well. So I also had a look at the distance of the region to the nearest downstream gene. So there's an understanding that although promoters and enhancers are generally upstream of genes, they're usually within a few megabases. So I took the distance to the nearest gene start, and again, I found a significant difference between the true and false positives, where the true positives were significantly more likely to be closer to the nearest downstream gene. So when I filtered here, I managed to get the ratio down to 1 to 29, but again, this is still very high. So the last thing that I decided to look at was conservation scores. So as I said earlier, sequence conservation doesn't always equal functional conservation, but conservation scores can still be useful, especially when these machine learning models are unable to currently deal with such imbalanced data. So I decided to add in conservation scores as an additional filter. So I generated a multi-species alignment with progressive cactus of all of the felid species that had a chromosome level genome assembly published, and calculated conservation scores using HAL-PhyloP. And this gave me a PhyloP score, which is a measure of sequence conservation, where anything above 0 is likely to be a conserved or constrained sequence, and anything below 0 is under accelerated evolution. And again, I found a significant difference between the true positives and the false positives. And this actually was the most effective filter. So I found here that the true positives were more highly conserved than the false positives. I managed to get the ratio down to 1 true positive for every 10 false positives. So this isn't perfect, but I wanted to double check that the model is still working. And even though I have a lot of false positives in my data, this isn't necessarily the model predicting incorrectly. It could be that these are real positives, but they weren't picked up in the ATAC-seq data. But to confirm this, I wanted to check that the model did look like it was learning real biology and really predicting functional non-coding regions. So explain generates a set of motifs that it's used to make these predictions. That's what makes it explainable. So I overlaid these motifs with the Jasper vertebrate database of transcriptional factor binding and found that there were many transcription factor binding motifs that were identified by the model that it was using to make predictions. CTCF was very strongly associated with a positive prediction. So when the model found the CTCF motif, it was more likely to predict this as a positive, which makes sense. And in fact, the only three motifs that the model was learning and using to predict negatives were involved in retinoid signaling, embryonic development, and centromeres. So it makes sense that these three functions are more likely to be associated with negatives because they're not likely to be in my training data. So my training data was from adults. I didn't include data from centromeres. So it's likely that this is why these are associated with negatives. But the outcome of this shows me that the model is learning real transcription binding behavior. So it's learning transcription factor motifs, different in each species, but because I'm combining these species, it's using all of this information to make its predictions. So this gave me confidence that the model is doing what I want it to do. It is predicting functional non-coding regions. So I then trained a model on all of the species and all of the tissues that I had available. So I trained an explain model with this and then applied this to the cheetah genome. So what I needed to do was break the cheetah genome up into 201 base pair windows. I did these with an overlap of 50 base pairs so that every site is covered more than once. And I only included the non-coding windows here. I gave this to the model and the model outputs a prediction score for each of these windows. And what I can then do is take the regions that it's predicting as as functional, overlay these back onto a cheetah genome and use this to annotate the functional non-coding genome in the cheetah. And then I carried out my filters to remove as many false positives as I could. So I took the non-coding windows, filtered for the explain score of over 0.75, and this reduced the number of windows I had drastically. I then looked at the distance to the nearest downstream gene start and took anything that was within 50 kb of this, and then looked at the median phylop score. So generally, a phylop score of greater than 2 suggests conservation or significant conservation. What I found here was that I only had 542 windows remaining after I did that filter. And I looked at ultra-conserved elements, and all of these overlapped an ultra-conserved element. So it's looking like the phylop score filter was becoming too stringent. So in fact, what I did was returned to the data set that had been filtered for the score and for the gene start site, and then took any that overlapped an ultra-conserved element. So I'm still adding in that filter of conservation score, but I'm not being as stringent using the phylop scores. So this gave me 292,000 windows that I think are likely to be functional non-coding regions. And just to check again that this looks like it's learning real biological patterns, I had a look at the transcription factor motifs that were found in these windows that were predicted as positives. So this is the plot here on the right, where I have the top 20 most commonly found motifs. And the color shows whether or not these were included in the motifs that the model was using to make its predictions. And the dark purple ones, as you can see, there's only 3 of them, are ones that the model was using to make its predictions. But what you can see is the model is finding regions with transcription factor motifs that are outside of just the ones it's learning. So it is learning some kind of architecture and structure of these functional regions and making its predictions on these, which again gives me confidence that the model is doing what I want it to be doing. So this is showing me that it is possible to predict functional non-coding regions in a non-model species using machine learning. But what I wanted to do next was apply this to conservation. So are there deleterious mutations in these regions, and what's the distribution of these? So firstly, I just looked at deleterious variants as a whole in captive and wild cheetah populations and did a general PopGen study. So I found my 65 premature termination codon loci, and I've also found my potential functional non-coding regions, which are windows of 201 base pairs. And I wanted to know how common are mutations in these regions. So this is the map I showed you at the start of cheetah distributions, and there are currently thought to be 4 different cheetah subspecies. It was previously 5. This is somewhat debated, um, separated by the colours you can see here. Um, and one thing to note is the samples that I had previously were from Namibia and Tanzania, which are now both considered the same subspecies. So I wanted to understand whether the mutations that I found in those individuals are shared outside of just that subspecies, so whether they're seen in other populations across Africa. So working with African Parks and the University of California, Riverside, I sequenced 2 South Sudan individuals to add to this population data, which is a separate subspecies and gives us an idea of whether these mutations are shared wider than just that one subspecies. And I also wanted to have a look at the captive breeding population. So whether the mutations that we see are shared in captivity and are being carried through with captive breeding programs. So I added in 30 captive cheetahs working with Klaus Copley at the Smithsonian National Zoo and Conservation Institute. And this gave me an understanding of the mutation shared in captivity compared to the wild. So I generated a VCF of these population individuals. So I included all of my novel sequencing and the 6 resequencing individuals that were previously published. So I carried out QC, trimmed and mapped, and then called variants, and carried out hard filtering using GATK. So I filtered for depth between 3 and 17, and this was the first and 99th percentile based on my data, and otherwise just used the recommended GATK hard filtering filters I checked, and all of these were suited to my data. And this gave me a VCF with 39 cheetahs and nearly 7 million variants. So I then filtered the VCF to extract biallelic SNPs only, remove the X and Y chromosome, particularly because I didn't have the Y chromosome to start with, but I removed the X as well, filtered for missingness. So anything that was missing in more than 0.15% was removed, in 15%, sorry, was removed. And then for each analysis, I did some specific filtering depending on the requirements of that analysis. So I removed SNPs with a minor allele frequency of less than 0.025, removed relatives based on kinship in the stub book, and I'll come on to that in a moment, and also did linkage disequilibrium pruning. So as I said, I did those depending on the needs of each analysis. But I started off with kinship, and what I found running kinship analysis was that my wild samples were unrelated to each other, which was great to see. So what you can see in this plot here is the top individuals are captive. Then we have the Tanzania and Namibia individuals here. These are Namibia, sorry. Then we have South Sudan and then Tanzania. But what you can see in these wild individuals is that they are all blue, so they are all negative or close to 0, which is suggesting that these individuals are not related to each other. But what you can see in the US population, the captive individuals, is that some of these relationships are closer to red, suggesting that there is some relationship here. So I had a look at these in more detail. I removed all of the other populations and just kept the US population. And what I wanted to look at here was we used some family groups in the sampling based on the stub book, and I wanted to just check that the genetic data agreed with the stub book recordings. And what I can see here is 4 main groups of highly related individuals. And in fact, this aligns with the family groups that we found from the stub book. So this confirms that the stub book has been well maintained, and it has accurate representations of family groups. So what you can see here are 2 offspring shown in the yellow family group, and then you can actually see the relationships with their 2 parents as well in this plot. Same with the turquoise ones, so you can see their parents here, and the green and the pink also. So these are all groups of siblings that are highly related to each other. Um, Sorry, one thing I should say here as well. So what we decided to do when we were removing relatives was where we had both parents included. So for these three family groups, we removed all of the offspring. And for this family group here, we didn't have the parents included. So we just took one of these offspring that had the highest coverage and removed the other one. So that was how we determined the related individuals to remove. We'd confirmed it with the stub book and with the genetic information. So I then had a look at overall population structure, and a principal component analysis here shows separation between Tanzania and South Sudan, but it shows that the US and Namibian individuals clustered together very tightly. It's likely that this is because many of the individuals that were originally sampled from the wild and brought into captivity were from the Namibian and South African populations. This is the largest population of cheetahs and is declining the least currently. Um, and historically, this has been where the captive population has been sampled from. So it makes sense that the captive population would cluster with this population. I then did a PCA just of the US and Namibian cheetahs, and the Namibian cheetahs do separate somewhat, suggesting that there is some relationship with the US, but there's quite a lot of diversity even just held within the US population too. So I also looked at population structure through admixture, and in fact, what I found was that the error value for a population of size of 1 was the lowest, so there is no population structure in these populations at all. And this is not unexpected. So cheetahs have very low genetic diversity. This has been found before. And I do also have very small sample sizes for the wild populations. So this makes sense, but this just confirms that although we see differences in the PCA, these populations don't separate that much. And again, you can kind of see this in the population differentiation. Um, One of the important things to note here is that the US is most closely related to all of these wild populations than these populations are to each other. So this is suggesting that there may have been some sampling from South Sudan and Tanzania as well into the US captive population, and highlighting here that US and Namibia have the least differentiation between each other, which again, as we've seen before, makes sense. So one of the things I wanted to look at was runs of homozygosity. So this is a measure of inbreeding. Higher numbers of runs of homozygosity and longer runs suggest more inbreeding. And what we can see is that the size and the number of runs of homozygosity in this plot on the left are higher for the South Sudan and Tanzania individuals and lower for the US and Namibian ones. And again, here, this is the fraction of the genome in runs of homozygosity, this plot. And you can see that South Sudan is the highest, and Tanzania and Namibia are also higher than the US. But you'll notice in both of these plots, there's one US individual that is an outlier here. So I wanted to have a look at what was going on with that individual. So I had a look at the total length of runs of homozygosity. I categorized these based on size and looked at these across all of the individuals. And this is the individual that was an outlier in the previous plots. So what you can see here is that this individual has a large amount of long runs of homozygosity, and in general, a lot more runs than the other US individuals. Otherwise, you can see that South Sudan is highest, followed by Tanzania. So this follows the same pattern that there is higher measures of inbreeding in these populations than in captivity. But one important thing about this individual is that I had a look at its pedigree, and you can see that there are basically cycles in this. So this individual, despite being part of the captive breeding program, has had a lot of inbreeding in its history in the captive breeding program. So all of these individuals, except these two wild ones, are captive cheetahs. And yeah, as you can see here, there's a large amount of inbreeding here and higher up as well. And interestingly, this was one of the individuals that we sampled for one of the family groups we were looking at. And we didn't know that the pedigree looked like this before we did this. but this individual has been included in a family group. So we can see its offspring and we can see whether the runs of homozygosity have been passed on. And actually, what we find is this individual mated with this yellow individual here. This was the mother of the offspring. And the offspring look typical for the rest of the population, despite the fact that one of their parents had such high levels of runs of homozygosity. These individuals look fine. So this is a reassuring result to find in that although some of the individuals in captivity have been exposed to a lot of inbreeding, the general measures used for captive breeding are just a measure of relatedness. And as this individual has been bred with an unrelated individual, it's not passed on much of this runs of homozygosity at all. And that's reassuring. That's showing that these individuals can exist in the captive breeding program and not pass on these very high levels of inbreeding. So once I had an idea of what was going on in structure and kind of levels of inbreeding as a whole, I then looked at deleterious mutations in genes. And I found highly deleterious single nucleotide polymorphisms in sperm-associated genes. So I ran tools called SNP-F and SNP-SIFT, and these score variants based on pathogenicity, so how deleterious the variant is likely to be. And I identified 684 variants with a high predicted deleterious impact. And I then took the genes that these variants were found in and did a Go enrichment analysis and found an overrepresentation of sperm-associated functions. So this again is showing that a lot of the mutations that we see in the cheetah genome are associated with sperm and could be causing the problems in reproduction and fertility that we see in cheetahs. And I also had a look at the distribution of these highly deleterious mutations across the population. So I've seen that inbreeding seems to be higher in the wild compared to the captive population, but I wanted to see whether these deleterious mutations were also fewer in the captive population or not. So what I looked at was the distribution of these deleterious mutations in the four populations, and then the proportion of the SNPs found unique to each population that were predicted to be highly deleterious. And what you can see is, although Namibia has a relatively low number of unique deleterious SNPs, it actually has a high proportion compared to the other species. So the proportion of the Namibia-specific SNPs as a whole that are highly deleterious is higher than the other species. So this is suggesting that although there's higher inbreeding in South Sudan and Tanzania, actually there's a higher proportion of deleterious SNPs in Namibia. And this is particular risk for the US population, particularly because a lot of the US population is sourced from the Namibian and South African subspecies. So these mutations could be carried through into the US. Although in the individuals that we've sampled, we haven't seen as high a population of these deleterious mutations. So finally, on to the ongoing work that I'm working on as I'm wrapping up my PhD. So I've spoken about the deleterious mutations we've found in coding regions and my work to identify functional non-coding regions. So what I'm working on currently is looking at deleterious mutations in these non-coding regions. So I'm using a combination of tools. I'm using NC-Boost that has scored variants in non-coding regions for pathogenicity, and this is based on human training data. And I'm also using a tool called LoadLift by Sam Speak in my lab. And this uses CAD scores and lifts them over from the human to the cheetah and scores variants as well. So I'm scoring all of the variants that fall within my predicted windows and then using a combination of Hi-C data that exists for the cheetah and also looking at the nearest downstream gene to these windows, associating variants with the genes that they are most likely to be impacting. And I am seeing currently a similar pattern in the non-coding mutations to the coding ones. So it will be interesting to see whether we see that there are a high proportion in Namibia as there are in coding genes, and it's looking like they're more common in sperm-associated genes as well. But I need to dig into this further. And some of the other things that I'm working on more into the future are collaborating with Heather and Klaus at the Smithsonian and George Mason University to look at the inheritance of mutations in the captive breeding program. So that's why we included the family groups in our sampling, and also looking at the distribution of mutations across wild African populations. And this is working with African parks. They're interested in what subspecies of cheetah they've got and what mutations these populations are carrying. So to summarize, I have identified premature termination codons associated with sperm and immune defects in the cheetah. I've demonstrated that you can accurately predict functional non-coding regions in non-model species, and identified highly deleterious mutations in sperm-associated genes that could have impacts for conservation. So overall, what does this mean? For captive breeding programs, it's interesting to be able to identify population-specific deleterious mutations and also consider the the combinations of individuals and whether this passes on things like runs of homozygosity. And this is important information for the organizations running captive breeding programs. For the wild cheetah populations, again, whether there are mutations that are specific to populations. One of the things that has been done currently in Africa to save some of the very declining populations is translocations of cheetahs. But if there are mutations specific to, say, the South African and Namibian subspecies that are then being moved up into a different population that hasn't been exposed to these mutations, we're just spreading those mutations further and they're more likely to become fixed. So this is really important to understand for conservation of wild populations, but also considering as well the genetic diversity of these different populations and whether this can be used to classify subspecies. But this isn't just important for cheetahs, this can be used for any non-model species. So the methods that I've used here aren't cheetah specific, particularly the non-coding work that I've done with machine learning. This can be used for any species that has an available reference genome. So this can be a really important resource for population genetics and conservation research. And finally, a human-centric impact. So I found a lot of mutations associated with inbreeding, particularly in sperm function, and these may be very relevant to human diseases and human fertility as well. So understanding these mutations in other species and doing a comparative genomics approach can help us understand the impacts of genes and mutations in humans as well. And with that, I'd like to thank my supervisory team, my group at the Earlham Institute, and my collaborators at Smithsonian Mason School of Conservation, University of California, and African Parks. And I'd be happy to take any questions.
Thank you very much, Jess. This was great. I must say it was a combination of, like brand new results, of course, but also PopGen lessons. I really liked it. And my questions actually reflect that. So I have questions that are coming from a place of curiosity because I don't do PopGen, perhaps unfortunately. So I do have questions about some concepts, but also I have questions more, I want to say, on a higher level, perhaps even philosophical aspect of when you touched upon, for instance, conservation versus function. I'm confident you're aware of the controversy around that connection. So I do have a few questions there. But again, thank you very much. This was great, very insightful, very well organized, and I learned a lot. So to begin with more conceptual questions, what are runs of homozygosity? I could have a sense of what they mean, but I think I was confused when you said, we measure the length and the number of them in a particular individual. So what are those?
So these are runs of the genome that have no variation in. They're homozygous
Okay.
consistently for a long run. So what this is showing is, a long stretch of the genome with no variation. If there are mutations in these regions, they are fixed and homozygous. And the longer the runs and also the higher number of runs suggests more inbreeding because there's less diversity, essentially.
Okay, thank you. And again, out of curiosity, what are the numbers in the pedigrees? Because the individuals were numbered. Do you have an idea of what what's the rationale behind the numbers? The
Those
600,
are just IDs to keep track of
just
the
like
cheetahs
okay.
rather than using names. They're just IDs. Everything in the stud book is numbered with an ID. So that's what those are.
And that's a good segue to my other question. What's a stud book? I did not know this term
Yeah.
actually.
So this is a document that essentially tracks every single cheetah in theory in captivity. Um, they exist for many, many species. The cheetah one is particularly well um, recorded and well kept up and maintained. But essentially, this has tables of all of the cheetahs that have ever been in captivity and then who the parents are and what organization is currently holding the cheetah. So from this, you can see familial relationships. So you can see for each individual if their parents are known. So if it's been part of captive breeding, their parents will be known. Um, and then you can track back that lineage. So every time a cheetah is born in captivity, it gets entered into the stud book, and you write down and you give it a new ID and you give the parents' IDs and where the cheetah is held, essentially. And the same for when a cheetah dies in captivity as well.
Really cool stuff. Yeah, I could sense it was something about it, but I found it interesting. Very cool stuff. Now, I think moving more towards the research, your research project per se, you mentioned that you didn't have the chromosome Y for your groups, right? Is that correct? Like you eliminated you did not consider chromosome Y for certain groups because for I think your resequenced genome you didn't have the chromosome Y. Uh,
Yes, yeah.
just out of curiosity, would you have any prediction of what the chromosome Y would tell us? Because I'm assuming, again, coming from a place of not knowing anything about chromosome Y, my understanding is that, it has very little recombination. I mean, if any, right? Because there's no homologous chromosome. It's like, so to speak, in its way to being lost. So what do you expect to find there when comparing
Um,
chromosome Ys of different populations, I guess?
Yeah, the problem with the Y chromosome is it's very difficult to assemble.
Mm,
So it's not included in a lot of reference genomes. Um, there are people working on this. Actually, Ellie Armstrong is working on getting more and more Y chromosomes for mammal species assembled and published. But I would expect to see a lot of these deleterious mutations causing sperm defects to be on the Y chromosome. A lot of the problems in cheetahs are male fertility, so it would make sense for them to be on the Y chromosome. Um, but yeah, they're just difficult to assemble, which means they're not included in a lot of assemblies. But I would love to have a look at the mutations falling
mm.
in that genome, in the chromosome. And I think, yeah, that would be a great kind of future project to look at.
Yeah, that's why I had that in mind because like you shown us, so much of the inbreeding phenotypic consequences are male related with sperm fertility. So I was thinking about that as well. Um, Now, so you've measured PTCs, which are premature stop codons changes in, which is one way of pseudogenization, right? So I think
Mhm.
this can become pseudogenes in different ways. Did you have a chance to look at other ways in which genes are being pseudogenized in these populations, or perhaps this future work? What do you think?
Yeah, so I think again, this would be future work. So one of the things I was looking at when I was starting that project was looking for a kind of genome-wide approach, including the puma in. So the previous studies had either looked at fertility-related genes to start with, or had done a genome-wide approach but not included the puma. So I wanted to see whether there was anything missing there because I could include the puma and look for cheetah-specific things. And this kind of led me to looking at the premature stop codons because I ended up finding more than I would have expected. So yeah, I only focused on the premature stop codons in the end and that avenue into pseudogenization because that is the way that genes become pseudogenized, likely because of inbreeding and low effective population size. Otherwise, genes can become pseudogenized if they are duplicated and one of the copies is no longer necessary and it kind of loses its function and it can accumulate mutations without a fitness effect. But because this wasn't so directly associated with the consequences of inbreeding, I wanted to focus on the stop codons.
Mhm, mhm. Wonderful. And I guess just to wrap things up, so you mentioned at the very end that having high quality genomes would be helpful as well, even in perhaps reclassifying subspecies. You did allude to how before we had five subspecies for cheetahs, now we have four, in fact. In fact, two populations that you studied, they have been grouped in one subspecies more recently. So how would a high quality genome help in the reclassification, if needed, of subspecies? I mean, very high level as well. Like I know there's a lot, high quality genomes are great for several things, but for that classification purpose, how would that be helpful?
I think the problem that I had in this study was I had very few individuals from each population. So it was looking like Tanzania and Namibia were quite different to each other, even though they're currently classed as the same subspecies. But I only had three individuals from Tanzania and four from Namibia. So I think in that case, having a higher number of individuals would actually be more useful. We've got a relatively high quality reference genome for the cheetah, but being able to map more individuals and also potentially long read individuals rather than short read sequencing that would cover the whole genome. We could have a look at variation in repetitive regions as well. And I think this would give us a better picture of the variation across the genome in these populations and whether they are significantly different to each other or not.
Well, really cool stuff. As I said, I really enjoyed your talk. I think I've interrogated you enough. So I mean, we can always chat more another time. But again, thank you for taking the time to teach us about your work, for bringing awareness to this important cause. I think anyone would agree that first we need to uh, take care of our species, and particularly such a charismatic and important species as cheetah. So thank you very much, Jess. This was great.
Thank you.
I will stop the recording now.